So I guess, I mean, what I wanted to do for the meeting today was just to run through some of the key action items from a meeting that was 2 weeks ago. Chemistry in the lab's been a little bit slow, of course, because Dana's just back with us in the lab, and we've been doing other things to do with some potency assays and toxicity assays. So I'll run through those in a moment, but because we have a special guest visitor, Flavio, I wondered if you would just say a word or two about how you came to be working on the series that you're working on, and I mean, a little bit of background, because it looks like we're working on similar structures. Did you want to just say a couple of words about the origin of the work you're doing?
Yeah. So we started working on with this kind of compound. So first of all, I usually work on heterocyte chemistry and related medicinal chemistry of heterocycles, focusing on new heterocycles or other opportunities we found. So we started working with around 2-3 years ago with these imidazopyridines, imidazopyrimidines, and trying to explore different types of functionalization. That was our main effort at that time. So CH activation, borylation, all sort of approach, and we applied this because I hired a postdoc that worked in Seattle with a group in the University of Washington, Michael Gelb, and he brought with him these structures that we were already working in our lab. So we put together the projects, and he was working with Leishmania and T. cruzi, and so we apply all those efforts to expand the library and the chemical space to find new active compounds. So that's where everything started, and since then we are becoming more productive, and we have a nice library of compounds. I don't know if I think Ben have already forwarded you, Laurie. I'm quite sure he had sent her the list of compounds, and those are just examples, because we have other types of functionalization and functionalized scaffolds that we have been working since then. So I think that's briefly says what we are doing, and related to the project I was just invited, and thank you for sending me the invitation.
Yeah, well, I mean, no, that's great. I guess your interest there is not in compounds that kill bacteria, then?
No, the first time, no. It was, okay. I was, in fact, I'm not involved in antibacterial until last year, when I started working, but with different compounds, different scaffolds, more natural product related chemistry, but it's an ongoing project, and right. So these compounds were not designed for as an antibacterial. So.
Right. I mean, and
do you know the mechanism of action in T. cruzi?
No, we are starting working with these with guys in Dundee. Yeah, there are some ideas, but nothing really well defined. So
okay,
it's main phenotypic based.
Yeah, we're trying to answer some of those questions ourselves, and it's currently, as you said, lots of ideas, but nothing concrete.
Yeah, nothing concrete. There are ideas on acting on proteasome that people believe they are the main potential targets, but I don't know. There are some doubts, at least from my side, about it. Okay.
I mean, one of the practical things we could do, of course, is given the similarities, we can try to exchange compounds, which will be very helpful. We can also, of course, try to make sure that we're not ever duplicating chemical space by sharing what we're doing. So this project is one of those where everything is fully shared and there's no IP taken. Sorry, there's no IP protected in the project, so it's all fully open. So, you know, in terms of the paperwork involved in sharing compounds and sharing data, there really isn't any. So it keeps it kind of simple. I don't know, it depends on your funding agency and what you want to do, but if you're also open to, you know, sharing compounds that we can, the structures of which we can reveal in the public domain, that would be very helpful. It depends how you work.
Yeah, well, I'm already working in the similar scheme with the Open Synthesis Network with PNDI. So right. It's working well. So we can share compound and we can spread the message open and freely with everyone. So no problem at all.
Right.
I checked the funding agency and there is no problem with them. In fact, what they want is that their name in the acknowledgements and say that, okay, it's I'm here, so, and I funded this project, and that's it. So it's up to me, and I think it's a good idea, and I advocate for this type of project.
Okay, great. I mean, as a result of the last meeting that we had, we're kind of focusing in on a scaffold that's slightly different from the one that we started with. Maybe it might be easier if I shared a screen, but we're in the middle of a lot of this right now. So this is from this is the meeting from today. These are the last data that we obtained for the potency assays, which were compounds that Laurie had sent us. These kinds of structures here. And, you know, in the past, we've found that for potency against MRSA, we need this 2-pyridyl motif. If that nitrogen is moved around the ring, or if it's removed, sorry, if it's moved around the ring, or if it's removed, we lose potency against the bacterium. But in the past, with other molecules that we've been looking at, I've got the same thing up here, a slightly different core, like this core here, like this, where we have a aromatic ring joined directly rather than through an amine linker. We have so far found that the activity against MRSA kind of tracks with toxicity in a mammalian cell line. But the latest data we have were that these compounds were reasonably potent against MRSA. And there were a couple of these for which Laurie had data which suggested that they were not toxic in a mammalian cell line. And so the action item for us from last time was to make sure that these 6 compounds, which we had evaluated in the potency assay, were then evaluated in a toxicity assay. And so the action item, I guess, is on you, Dana, right, to see if Paul Stapleton still had those DMSO samples that we could send over to Alex for evaluation.
Yeah, so I hadn't emailed him yet because I couldn't remember which ones and I hadn't gone back and checked. So we want all of these reevaluated in Alex's assay.
Yeah, I think so.
Okay.
And you know, we had a lot of discussion last time about the importance of making sure that there are, if we're using different potency assays, that we have, you know, so-called bridging compounds that that are evaluated throughout these different assays to make sure we can compare. So it may make sense, depending on what Alex's plan is, to include one of our other standard molecules that we've already evaluated in something else, in case she decides to change the assay. I mean, I'm very happy for her to keep the assay exactly as it is, just to make sure there isn't another, you know, variant. They came at this whole thing from a different perspective, of course, because they want to find cytotoxic compounds, and they look at how the cell dies to try to reveal something about the mechanism of action. We don't need that yet, I don't think. We just want to find the selectivity index. So I'd be very happy if she just carried on the way she was going before. In which case, these 6 and maybe one of the controls that we had from last time that she evaluated would be enough.
Matt,
yeah.
is it worth looking at some of, is it worth actually asking DNDi if they would look at some of your compounds in the assays that we're getting tested consistently as well?
That's the MT something assay.
We've got MRC5 and
MRC5, that's it.
there's another cell line that is eluding me right now. THP1?
I don't know. I can't. I'm not well versed enough in the differences between these assays to know if we need more than one or what they're telling us, other than the fact that they are toxic or not.
Yeah, so I guess my thinking on that was really more about if we're going to continue to send compounds, well, I intend to continue sending compounds to you that kind of go along this vein. And so to actually have a point of reference of what the activity for your compounds are in their assay, and then we can readily compare anything that I have sent you across, because you will know kind of,
that's true.
Just like benchmarking it in the assay that we're using, and we get the data on.
So basically, yeah, the compounds that we had evaluated in the tox assay here, just send them over to you.
We could ask the NDI if they could do that. And I mean, it's what, 6 compounds, I think.
Yeah, I mean, these are the ones.
It'd be useful to have, all of them in all assays.
Mm.
I mean, you already have the data on these guys from them, right? I guess the ones that
And I'm pretty sure I've shared that with you, and if I haven't, I can do that. Yeah, Dana's nodding, so I have. Good.
That would, no, that'd be very nice. I guess, yeah. So the ones that Alex evaluated last time, it would be good to have them done in that assay as a cross check. That would be very helpful. Yes. Okay. So Flavio, these structures, I mean, these are presumably quite similar to the ones you're looking at. Do you see the obvious similarities? I mean, do you? I'm not quite sure what you have lying around in the fridge there, but do you have variants of these kinds of structures?
Yeah, the first thing I can see clearly is that I have similar compounds that substitute we have a spacer between the imidazopyridine ring and the amine that is substituted at this 3 position. I believe it's the one at the side of ortho to the pyridine ring. So a CH2, a methylene here, giving a spacer between the amine, the aniline ring, and the imidazopyridine ring. I don't know if it could help to expand the expand the chemical space and give you insights on how this amine influence the activity.
So there's a CH2 in here.
Yeah, exactly. In at this point, it's easy to make like many reaction. It's very simple, and we can use them. We can also alkylate the nitrogen and have similar compounds with a spacer between the aniline and the imidazopyridine side. And we don't have any pyridine substituted, but it's not a problem to prepare those, as it seems to be important to the activity, So, and on the pyridine side of this ring, imidazopyridine, have you tested any kind of substitution? Any substituted ring at this side?
I think some methoxys in this position have been made, and fluorines, I think.
Okay.
But not hugely. I think that Laurie has looked at some of those things, but not hugely. No, I mean
Laurie say that there's some issue with toxicity with some of those substituted compounds.
With the methoxy there, yeah.
Yeah, we've seen a differential toxicity profile between the 6 and 7 positions of that.
Okay, and also in the pyridine ring is also substituted rings at this position? Because you've mentioned that you have nitrogen around the ring, and this affects the activity. But what about the substitutions? What about?
Yeah, Dana, forgive. I think I'm right in saying we haven't really explored that, right?
Yeah, we haven't. We've put heterocycles out there, but not substituted ones.
Okay.
Yeah.
Because this gives us opportunity maybe to prepare something and try to understand better the influence of the pKa of this nitrogen. Because if it's maybe, I don't know, if it's needed to be at this position, maybe some.
Well, we don't know exactly for these N-alkylated compounds. We've never made an N-alkylated compound with the bottom pyridine ring changed.
No, but we haven't even really put anything on the pyridine, right?
That's one of the areas that we're actively exploring at the moment. I need to touch base with Quillen, who's doing doing all of the chemistry at this point on this series, and just see. So we were, yeah, if it makes sense and you're interested in testing them, I can certainly send some your way, because we're supposed to be shipping out next week anyway. So we could just.
Yeah, cool. That would be great.
I'll share a Smiles file or something so that you can see what we've got.
Thanks. Great. And Flavio, I guess I had a couple of questions. One was about the this 2-pyridyl motif. You know, in your case, do you also need that? So if you move the nitrogen around, do you see a drop off of potency?
Not at all, and we don't have many examples on this series, but it doesn't seem to be very important for us. So
okay,
if you change this by other heterocycles or benzene, we don't lose activity. So it's not the most important part of the molecule in our case. So I think we can stick to this kind of substitution. Then I think the important question here, if it's important, the position of the nitrogen in the pyridine related to the imidazole and pyridine. I don't know if you have any idea why.
We sort of guess that that's a very good metal binding motif.
That's what I was up to say. Because it can complex very, and so maybe it's important to have a very basic nitrogen in the pyridine ring, and then increasing the basicity of this or the electron density at this position. That could be nice. So can think on different substitution, but Laurie is already working on it, so we can see the results and discuss a bit of it.
That's good. Yeah. Okay. And obviously, I mean, it may be that, of course, we're looking at a completely different mechanism of action. I'm assuming in your case, you were able to identify compounds that were potent against the pathogen, but not toxic.
Yeah, we have some that are not toxic, and if needed, we can send for those that are interested. We can send list of toxicity.
What if we just cross-pollinate and say, we'll send compounds that are potent against MRSA, and you send us compounds that are potent against Chagas, and we test Chagas compounds against MRSA and the MRSA compounds against Chagas?
Yeah, sure. I guess I think that we probably need to limit them to compounds with this motif. Otherwise, we're likely to, well, there's a possibility that we're moving away from the mechanism of action that perhaps we're involved in here. It feels like we should maybe limit it to that. Things that are synthetically demanding to do for the first time, like put a CH2 linker in between here, would be great to evaluate those rather than have to make them from scratch. That would be really a time saver. And vice versa. So, you know, all of our compounds are listed on the Google sheet of compounds that we've made for the project. So they're all available, and we probably have samples of most of those, I would guess. So yeah, similarly, you know, if there are particular motifs that you're interested in, Flavio, that we could provide you with compounds for, then that's absolutely great.
Great, that would be great. And I can do the same for sure.
All right, great. I mean, it is. I mean, compounds with this 2-pyridyl motif, with the spacer between the nitrogen and the ring, there would be very good. And Laurie's compounds with substituents here would be. I mean, that would be amazing. That covers a lot of ground, actually. And yeah, so that's the main thing, along with the toxicity evaluations of these compounds. Those are the main things, I think.
Did you ever make any benzimidazoles?
Yeah, we have some. We have some benzimidazoles without, but I don't know if you have this pyridine substituted benzimidazoles. Yeah, I don't know, but this is something interesting that
Might be a
could
good way to segue into the
maybe.
Yeah.
Okay, good. Dana, things have been busy, but on all fronts. Do you have any chemistry update you wanted to talk about, or not?
Sure. Yeah, I am working on the benzimidazoles. So my big issue that I'm having with these compounds is purification, and I'm having to do at least 2 columns to get anywhere near clean. So the chemistry is fine, and then it's the purification that's kind of a pain, but I've got a few that are close. So a couple that I've made with the one pot that I think probably are just sort of filling in some SAR gaps, and then a few of the benzimidazoles that I'm hoping I can get clean so that we can test them too and test that scaffold. So I can share structures, but that's the basic summary. Yeah.
Okay. I mean, you don't happen to have on your screen the benzimidazole that Chris was talking about, do you?
Yeah. So the top right.
Great. This was not a prepared thing, by the way. I'm asking Dana Cole. I didn't know she'd have this, but she's prepared.
Just in case. I made it just in case. Right. So we're looking at the chlorine. Oh, sorry, that should be in our group. So the chlorine on the core, which we were sort of wanting to explore as well, and then potentially seeing if we can substitute there. This was an analog that we'd proposed, which is sort of a crossover between one of the ones that Laurie sent us that was active with the amine in the core, and then these as well. So the paramethyl and the parafluor were active in the original core, and then some of the benzimidazoles with these substituents that showed activity.
Dana, have you tried reverse phase purification? That's what we found works best for the bottom set.
Yeah, well, I'm doing a normal phase and then a reverse phase. Not HPLC though, because I've just been running a reverse phase on the biotage. So I mean, maybe
that's how we're doing it.
Prep or something.
That's how we're doing
yeah.
it.
Okay.
Just but we don't bother with a normal phase first.
Normal phase
reverse phase.
Yeah.
One
so I'm still
phase
getting some pore separation doing that.
Okay.
Or there's stuff in the NMR I can see. So yeah, unfortunately.
Okay.
Recrystallization?
Trying it. It helps, but there's still some stuff. And also our LCMS is down at the moment. So there's a couple that I do want to check that I think might be clean enough, but I'd rather go by LCMS and NMR on that. So I'll check those.
Okay, great. Thanks. Okay, and then I just wanted to mention the stuff that Giada was doing. I didn't ask you to prepare anything, Giada, so I'm not asking you to present big time. But you've been looking at structures that may be relevant to us in the sense of metalloenzymes with things bound in the active site that look like the 2-pyridyl motif, and trying to find examples of that kind of behavior. Did you want to say anything about that? Because it's very much a sort of ongoing search, because it's sort of part of your your research project is to look through some of those things.
Yeah, I've found something interesting regarding methionine aminopeptidase, because I found that structure similar to our compounds that has the core with the two pyridyl motifs is really similar to another structure called, another drug actually called thiabendazole, that chelates the metal ion present in methionine aminopeptidase. And so I tried to do the docking experiments with this target and our molecules, but it seems that doesn't work because it seems that the core, it does not interact with the metal ion, but the other part of the molecule, so the benzene ring with the fluorine, will interact with the pocket and not our scaffold. But yeah, but it seems really interesting because the structure of this drug is really similar to our compound. Yeah, if you want, I can share this. Sorry?
I also found another possible hit on Twitter. I did a simple search and found something. An enzyme in staph called STP1, serine threonine phospho something or another 1, STP1. I think I mentioned that. There was an inhibitor of it in humans that looks almost identical to our drug. It's called STP1. I have it on, not logged into Twitter right now, but it's STP1 and MRSA, and it's
That's useful. I mean,
serine threonine phosphatase.
Okay, that's useful. So, I mean, I think that Giada's at the moment also learning the use of some of the software here to do some of the docking experiments, and this is a useful segue into that. I mean, ideally, I guess I was interested in if we're talking about a compound that is toxic by virtue of chelating a metal ion in an enzyme, what is the classic case of that? What should we be assuming is the mechanism of action? And I didn't know, so I sort of instructed Giada to run off into the wilds of the chemical literature to sort of see if there was something there that we could use as a starting point. Obviously, if it's MRSA relevant, that helps a lot. But if there is a known target which makes things toxic, we should know about that. And I don't know what that is. And if you happen to know, anybody else?
I guess anything that interacts with the metals has potential for doing that. I mean, it could affect things like, electron transport chain, lots of things.
Yeah, because it could be not an enzyme mediated thing. Absolutely.
Yeah.
What was the name of the drug that you said you were docking against? Like, what was the name? Do you know?
The name, you mean, if you want, I can share something if you want.
Yeah.
Sure.
It isn't, I haven't prepared something really
I didn't
good,
warn you. Sorry, I'm just picking on you.
At least you can see the structure. Sorry for this time.
Getting there.
Okay. Okay.
Is it loading your lab notebook?
Yeah. But if it doesn't work, I will. Okay, it doesn't work. I'll send you something different.
Okay, that's all right. I mean, you've got it on you're assembling a GitHub issue on all this stuff anyway. But maybe, yeah, you could just ping Laurie with that drug name.
Yeah, so the compound name is thiabendazole. And
Okay.
if you want, I'll send you the paper in which I found the similarity between. No, I found the docking part with this drug and the target. Yeah.
Okay, great. Thank you. Perfect. I was just going to mention one last thing. I mean, so obviously Giada is carrying on with that work, but it's work in progress, which is good. So we'll keep on touching base on that. The other thing I was going to mention is the Haifa guys have delivered their scale up. What's the word? Broth, I suppose, mixture of their metabolic stew of our compound. So they've done their SIP screen, and they've scaled that up, and they've shipped us a metabolite, which Dana at some point has to try and purify. And it's going to be, it has I think a shoulder on the peak. I think that's one of the things. But when we've got the prep LC up and running, we will fish out what that metabolite is in sufficient quantity, I hope, that it will give us a structure for the main metabolite. So it's sitting there in the fridge or the freezer and ready to go. So that's a really nice input from them. Obviously, it's on one of the earlier compounds that we were looking at, but nevertheless, I think it'll be a very nice bit of extra stuff for our study. That's just an update, and obviously it's on us now to purify and analyze that. All right, that was all I had actually for today. So I think, but I wanted to ask if anyone else had anything and I've missed anything.
What's the best way to share the structures and then the compounds and how to ship it? And
yeah, I don't know really. I mean, if you have a spreadsheet of all of the compounds, then of course we can just put that up onto GitHub, which is the platform we use, and post it there so we can search it. If you'd rather do the search yourself, then we can send you some smile strings of the most relevant compounds, and we can see if we can figure out what's most similar. It's really up to you. I mean, the best thing is if you were able to post some things yourself on GitHub by getting an account. We all have these accounts, and it's a very sort of not spammy platform. It's very easy to use, and we can direct you, where to put things. And that keeps everything transparent and public and allows you to control it. If you're happy to do that, then we can just give you some advice about how to do it. It's really easy, right? I mean, everyone here uses it, and it's we're not geniuses. We're not software people. Well, Chris is a software person to some extent, right? But none of the rest of us are software people, and we use this stuff pretty well. So it's pretty easy to do. So if you had things you wanted to post and thoughts you wanted to, and you were happy to get involved in the discussion, then it would be great if you could post it directly. So we can guide you on how to do that.
Yeah, that's great.
Okay. And
to have some ideas, and would be great to discuss with you.
Okay,
that'd be fantastic.
I mean, if you want, the first thing in that case would be to sign up for GitHub and send me your username.
Mm-hmm.
And then we can take it from there. That'd be the easiest thing. All right.
Great.
Fantastic. Great. Well, if no one had anything else, and if we'll assemble some action items on the issue from today to make sure that everyone knows what we decided, then we're all done.
Just one thing, I know on the indexing compounds on the getting Google to index compounds, I noticed that Mike has managed to find a way of doing that. It requires a bit of manual intervention at the moment, but it looks like it may be a good approach. Okay, great. I got the sense from the correspondence that that's what was happening, but he sounds like he's nailed it. Yeah, I mean, basically he's mirroring the sheet. So somebody will have to regenerate that periodically. Right. But but it works anyway.
Okay. All right. That's wonderful. Okay. Well, I mean, it's very nice of him to do that. Mike is a long-standing supporter of the technical side of everything we've been doing, and he comes up with solutions like this. So that's really good.
Yeah.
All right. Wonderful. Nice to see everybody. Thanks for coming along.
All right. Great.
See you
Thank you.
next time. And really nice to meet you, Flavio.
Thank you. Thank you for the
Right.
opportunity.
Right. Thanks. Great to meet.
See you.
Bye.
Bye.